Steve Reeser is next. Most of you are going to know Steve, but he worked with Virginia Department of Fish and Wildlife. Steve is talking about the application of eDNA sampling along with standard electrofishing methods to improve management of wild brook trout populations.
Thanks, Nat. Thanks everybody being here, all you in virtual land out there. It's great to see everybody. Yeah, it's great to be addressing the joint venture. Just a little side note, I would think it was 2004. Myself, along with Jerry Koza with Fish and Wildlife Service and Matt Besler with North Carolina, we got together to plan the initial meeting. It was held here at NCTC, and I think that's also where Koza came up with the name for this fish habitat partnership, the joint venture. That's Jerry Koza's work there. But and I want to acknowledge my author here, Brad Fink, who's our cold water biologist. Brad was here yesterday. Unfortunately, couldn't be here to give the presentation. I think he may be on the virtual end there, but wanted to put a shout out to him about putting this presentation together. Hopefully, I don't botch it too bad, Brad. But this is probably a great transition, this talk, for after lunch. And it's not going to be, it's not data heavy. There's no graphs. There's no statistics. It's pretty straightforward about what we've learned in Virginia with, applying the methodology of environmental DNA monitoring for brook trout management. Next slide, please. Oh, do I have the?
Other button?
Well, I'll try and. Oh. Just want to mention my co-authors there from the Forest Service that did some of this collection and some of the analysis as well. But so the background, Virginia, we've got a pretty robust wild trout resource, over 3,000 miles of stream. That's in our database. We have over 700 wild trout streams identified. That's brook, brown, and rainbow. There's about 2,500 that are allopatric brook trout. Next slide, please. Okay. So, how has the Virginia Department of Wildlife Resources monitored these wild trout populations? Well, going back originally in the 1970s, our agency did a comprehensive trout stream survey of the entire state of Virginia, built that database, what that resource looked like. We collected some information from anglers, angling pressure on wild trout populations, and that was in the early 2000s. We built a cold water stream classification system back in the 1970s. There I mentioned we had that cold water stream database where all of our data lies. And then part of our monitoring program for that wild trout resource has been going back to those streams that were identified as wild trout on a 7-year rotation basis. And then we have done some repatriation, restoration of wild brook trout populations, and it was Matt Culp's talk mentioned some of that work there. Next slide, please. So get a little background here on the environmental DNA work that's been done in methodology, testing the accuracy. The two pivotal papers that are out there, one by Wilcox and Company, and then Baldigo. One was the out west, Wilcox's paper, and Baldigo was the work that was done in New York State. Very comprehensive, testing the accuracy of picking up brook trout DNA in streams. And so we've gone with some of that, and I'll mention in the presentation here, with the detection level of brook trout at a 1 kilometer scale, stream scale. Next slide, please. So our objectives were to compare our standard electrofishing protocols that our agency has used to monitor brook trout populations to eDNA sampling accuracy, and that helped go back, and I'll talk a little bit here about how looking at determine if our database records that were indicating where we've lost brook trout populations, how accurate that is, or is that correct information, and to examine where repatriation may be needed to improve brook trout resilience to climate change. Next slide, please. So our standard electrofishing backpack electrofishing protocols, we select a representative reach, no culvert pools here. That's one of our criteria, and the minimum of 100 meters. So we have approximately 100 meters. We'd like to have one good riffle run pool complex in that site. All fish are collected. We measure and weigh all trout and game fish. We count and aggregate weight for all non-game species. And then we're obviously getting some stream length and width measurements. The eDNA sampling protocol, we followed the Rocky Mountain Research Station's protocols. That's who funded this project. And I mean, this is what I wanted to say at the beginning. I kind of jumped over there a little bit, but it was at a Brook Trout joint venture meeting, I believe, in Rhode Island, where I think maybe it was in a break, was talking to Jake Rash, North Carolina, and Jake said, "Yeah, I think the Forest Service is looking to, do some eDNA monitoring for brook trout in the east someplace." And so that's how I got in contact with Ashley Walters with the Forest Service, and put this project into motion. But so we chose appropriate sampling sites in part of that, and I'll talk a little bit about how we chose those sites. The protocols here are filtering at least a minimum of 5 liters of water for improved results, and then we were obviously labeling our samples. We were freezing them within 24 hours after collection. And I will mention that in some of the analysis that there were 3 markers. I guess that's the right terminology. Jason, you can jump in there, but 3 markers used for brook trout. And so the data, I'm not going to show that, but if any one of those 3 markers was detected, that it was a positive for brook trout. Okay, next slide, please. So in 2019, working with Ashley Walters, she said, well, where do you want to do some sampling? There's basically wanted to populate their database in a National Genomics Lab database. And so they didn't give us any direction on where they wanted to do those samples. So we went in, and there were 36 streams in our database where we had identified through presence and absence brook trout being present through electrofishing at 1 or 2 samples pre-1990. So it might have been in the 1970s and in the 1980s. And then we came back to those same locations, those same streams post-1990 through maybe 1, 2, or even 3 sampling dates, periods, and we did not collect brook trout. So those were candidate streams we wanted to go to and do the eDNA monitoring to see if we truly had, those populations had been extirpated. And then because of what the Forest Service said, well, we'll fund up to 80 samples. So we said, well, we'll pick 44 additional streams, and those were not, they were just kind of, some of those streams were ones that we maybe had never surveyed, or we had surveyed with electrofishing and not found brook trout. Some I knew there were brook trout there, some not. So it was just kind of a mishmash of those. And then in 2020, we did a study where we paired electrofishing and DNA, like eDNA monitoring at the same sites. And that's where we arrived at the site on the stream. We collected our eDNA sample, and then we immediately electrofished from that point upstream 100 meters minimum. And then, so they were paired 41 stream sites with eDNA and electrofishing sampling in 2020. So some results, like I said, there's not some fancy results here, no statistics involved with it, with the analysis, but so in 2019, out of those 36 streams thought to be extirpated, with populations extirpated, 8 streams had positive brook trout DNA results. The 28 streams that were marked for possible repatriation. So that's kind of, it's pretty much, the 8 streams there were ones that jumped out as there we would have missed with our electrofishing using as, our sole method of detection. And then we obviously, recognize that water quality variables need to be examined. What's the cause for that extinction? Next slide. So results from our 2020 paired electrofishing and eDNA monitoring results. So a total of 41 different stream sites were paired with our standard electrofishing protocols and the eDNA. And so I go back, I mentioned that those protocols were developed in the 1970s during our statewide trout stream survey. All 41 of the sites were positive for eDNA brook trout. Only out of 37 were positive electrofishing samples. So that's, there again, it's just a handful that we would have missed with electrofishing. And I have a little asterisk down here that in 2020, the U.S. Forest Service, the CAT crew out of Blacksburg, went back and resampled 40 streams at the same sites that we had sampled in 2019. But what they did in 2020 was they went upstream from that initial sampling site 1 kilometer, eventually because they ran out of stream, to kind of, test that hypothesis that the other researchers have found with the accuracy of 1 kilometer. And the results kind of from that, kind of validated what's published out there as far as the accuracy. So discussion, determining the presence of absence of brook trout using eDNA seems to be more accurate and efficient than our standardized electrofishing protocol. That was pretty apparent. And so our agency moving forward plans to incorporate eDNA as a component of our standard electrofishing protocols. And the ability to sample accurately and more frequently will assist, with, reaching the goals outlined in the joint venture, as well as the Chesapeake Bay agreement, the brook trout action goals there. And I think, on a local level in Virginia, it's also going to help us identify maybe tributaries of streams that are brook trout are using seasonally. And so that where they're maybe outside of the framework of regulatory protection now, our state sister agency, our regulatory agency, Department of Environmental Quality, recognizes, we have special protections for time of year restrictions and other things for wild trout streams. And so we may be able to add some more of those into that regulatory framework for protection. Next slide, please. So we found that the eDNA sampling can be a useful tool to determine extirpated populations and where repatriation may increase resiliency of brook trout in Virginia. And it's going to allow us to efficiently monitor potential population loss due to pending climate change. And I put down here this last bullet, the future potential of eDNA here. I know there's researchers that are out there actively looking at this as where we can maybe get some quantitative data on populations from filtering water. I don't know how close we are to an accurate method for that, but I think that's certainly on the horizon. And then, limitations with eDNA monitoring, I think from my perspective is for as far as that spatial occupancy, the downstream extent. We talk a lot about that, of understanding what that downstream extent is of these brook trout populations. And a lot of it's driven by water temperature in most cases. But with 1 kilometer of the detection limit, it's really not going to be useful. We're still going to have to go in with visual, whether it's snorkeling or electrofishing, to really determine what that downstream extent of those populations are. And with that, I'll take any questions anybody has.